So in this unit, we'll be talking about how cells divide. And so to talk about that, we first have to talk about something called a cell cycle and talk about how the genome, so the DNA inside of cells, duplicates itself in order to get a copy into each and every cell in your body. So the cell cycle is basically the life cycle of a cell. So cells have to go through a series of steps before they divide, where they grow, they develop, they do all their normal processes, as well as making an extra copy of DNA so that when they split in two, both new cells have their own copy. So some of the steps that a cell has to go through before it ever thinks about dividing happen during a phase called interphase. So on this image here, we can see the interphase actually takes up most of the cell's life, right? So we have interphase happening before we ever actually start dividing the cell. It's basically a quiet phase between cycles of division. So before a cell decides to divide. Most of the cells spend the majority of their time in interphase, making proteins, doing their stuff like your muscle cell, you're contracting, you're in interphase. The mitotic phase, or M phase, is a relatively short period of the cell's life. So here we see the M phase, during which mitosis happens. So mitosis is going to be the process of actually dividing the cell into and parceling out those two copies of the genome that we make into each cell. Okay, so interphase itself is broken up into three parts because we need to have a couple of different things happen in order to be ready to divide if we decide that we're going to divide. Okay, so a non-dividing cell lives in G1 or something called G0, but we're not going to get into that because what we're really focusing on here are the steps that are going to lead up into division. So if a cell is going to eventually divide, it's going to live in G1, at which point we have genes being read, so we're making proteins, we're expressing traits, we're doing stuff. The cell is doing stuff. Now, once that cell has decided that it's going to divide, we need to make an extra copy of the DNA. And that happens during our next phase. So S phase of interphase is where something called DNA replication happens. So in DNA replication, we're going to make a copy of the DNA. We're going to talk in the rest of this lecture about how DNA is packaged into structures called chromosomes. So we're going to copy each of those chromosomes in S phase to create something called sister chromatids. And we'll get into what that means in a second. After S phase, there's a final phase of interphase called G2 phase. G in all of these actually stands for gap. So it's a second gap phase. And this is where we're preparing for cell division, doing things like making extra organelles or something. So making sure that the cell will have all the structures that it needs in the new cells, and also basically storing up some energy because cell division actually takes a bunch of energy for the cell. It's kind of hard. Okay, so interphase is G1, where we have the cell doing its stuff. S phase, where we copy the DNA, and G2 phase, where we wait, kind of rest up, get ready for mitosis. Then M phase is the process of mitosis, or actual physical cell division, physically pulling things apart. And we'll get into that in the next lecture. But before we do that, let's talk about the DNA replication that's happening during S phase. So DNA is double-stranded, so we have two strands, right? And we're going to use each of those two strands, those two complementary strands, we call them, right? They're complementary because they sort of pair up. Okay. So each of these 2 strands is going to get used to make a new copy of DNA during DNA replication. So what's going to happen is we're going to take our parent molecule. So in biology, when we have something that makes a copy, we'll often call the original the parent, even if it's a molecule, not like a person. So we have our parent molecule with its 2 complementary strands. We're going to separate the strands, and then we're going to use those separated strands as templates and pair up our bases, a new strand with a parent strand. So we call the new strand a daughter strand. So we have our 2 parent strands, they split apart. So then we have a parent and a daughter. And over here we have our other parent and our other daughter, which is eventually going to give us 2 brand new double helices. So 2 copies from that original single double helix. Now, this process is regulated by enzymes. So there are a bunch of enzymes that are basically, you know, like our construction vehicles that do the job. So we're going to go through them, hopefully relatively simply. I'm sorry, there are a lot of them. So the first enzyme is called topoisomerase. Now what topoisomerase does is the DNA is in a double helix, so it's all wound up. So it's basically kind of hard to split those 2 strands from each other while we're all coiled. It gets kind of messy. So topoisomerase basically straightens up little sections of the DNA so that we can start to pull the 2 strands apart. Helicase is the enzyme that actually does the pulling. So helicase breaks the bonds between our complementary strands. So it breaks the hydrogen bonds connecting our nitrogenous bases. So here we see topoisomerase. It's around our whole double strand. It's uncoiling the DNA. And here we see helicase, kind of like a zipper, just going and breaking those hydrogen bonds. We also have some other proteins called single-strand binding proteins. And their job is basically to prevent our 2 strands of DNA that we've just opened from snapping back together. Because once we break those hydrogen bonds form because of chemistry, right? So after helicase is gone, they would want to just like right back into place. We don't let them. We cover those nitrogenous bases over with single-stranded binding proteins while we wait for the next part of replication to happen. Okay, so at this point, we've opened up our DNA. We actually do this in a little bubble called a replication bubble. DNA is really big, really long, so we don't do the whole thing at once. We kind of just like have a little pocket that's moving, allowing us to do this. Okay, so what's happening after we open up our strands in our little replication bubble is we need to actually start laying down our daughter strand, so the new DNA. A funny thing about DNA in DNA replication is you actually can't just like magnet on nucleotides automatically. It doesn't work that way. We need enzymes to kind of link them together, and we actually can't just place a piece of our first piece of DNA down. DNA nucleotides need something to stick to. They basically have to hold hands with other nucleotides. That's how we stick them on. So before we ever start putting down new DNA, we need to put down a primer. So our primer is just going to get it started. We're going to delete it at the end, but we need it to start sticking on DNA. So we make our primer out of RNA. So we have a little RNA primer laid down by our enzyme primase. So here in the image in yellow, we see our RNA primer. So those are some RNA nucleotides that we stuck on. And here's in orange primase laying it down. Okay. Once we have a primer and we have something to stick our nucleotides onto, we're going to have our molecule DNA polymerase 3. It's going to latch onto the primer and then start pulling in nucleotides, pairing them up, A to T, C to G. So here we see another primer that has been laid down here. And this sort of thumb structure, that's DNA polymerase 3. and it's laying down new DNA nucleotides. That's what we see in blue here. Okay. So after we've laid down new DNA, we're going to need to go take out those pieces of primer, that RNA that we laid down to start off. We're going to need to delete the RNA and then seal up the holes, basically. So we're going to need to remove the RNA because we want two new DNA molecules. We don't want like some mishmash with random amounts of DNA, random amounts of RNA. That would not lead to good results when we start to try to copy genes and express traits. So DNA polymerase 1 goes through, it pulls off those pieces of RNA primer. Now we can lay down some little nucleotides where the primer was because we have stuff to bind them onto because we've already laid down DNA. So there are DNA nucleotide neighbors that we can put covalent bonds onto. And ligase is basically going to seal up these holes. So if we had our primer, right, we had our new DNA here, we pull off the primer, we put on some DNA here, we're going to end up with a hole basically between neighboring chunks. We're going to seal them up with ligase. So that's a pretty basic process for copying either of our parent strands. But a little bit of a complicating factor is that DNA strands are anti-parallel. So if we have a double helix, one of them goes 5' to 3' this way, and its complementary strand goes 5' to 3' this way. When we're copying, so we have topoisomerase unwind, we have helicase break open our bonds. Helicase is going to keep unzipping basically in this direction, opening like that. DNA polymerase 3 can only work 5' to 3'. So on one strand, it's going to be working towards the fork, this little corner that's opening up. So it's going to be working in the same direction as helicase. So it can just work uninterrupted because every time it lays down nucleotides, we're opening up more and more DNA for it to run down in front of it. And that's going to be our leading strand. On the opposite strand, we're going to have basically DNA polymerase laying down DNA in this direction, but the fork is opening behind it. So as it moves forward, suddenly more DNA is being exposed behind the spot where it started. So it basically has to double back and redo that little bit that has now been exposed. It's going to have to jump back every time helicase opens up more DNA. So that strand with the little different fragments, the little different chunks caused by the doubling back is called our lagging strand. And the little chunks that DNA polymerase 3 is making are called Okazaki fragments. So at one strand going towards our replication fork, so towards that corner in the same direction as helicase is our lagging direction. But on the strand where we're working away from helicase, so away from the corner, away from the side that's opening up, we have our lagging strand, and it has to make the new DNA in chunks. And that part where we make it in chunks is really where ligase, the enzyme that sticks together little gaps, becomes really important because we're going to need to link those Okazaki fragments back together. And they actually each have their own little RNA primer because that whole process that we discussed on the last two slides has to happen for every little fragment. Okay. Moving back to our idea of the cell cycle, this whole DNA replication thing is happening during S phase of interphase. So we had G1, S phase, G2. At the end of S phase, we've copied all our chromosomes. So we have homologous pairs. We copied one of our pairs. We copied the other of our pair. So we have two identical copies of every chromosome. And we're basically going to keep them kind of connected at the center, these two identical copies. We're going to keep them packaged together so that as the cell divides, it can move them around together and sort them out really well and make sure that one identical copy ends up in one cell, and the other identical copy ends up in the other daughter cell. So after S phase, where we have these two identical copies, and we keep them kind of connected in the center, that's why we'll often see our chromosomes drawn as X's. That means we have two copies of the DNA still connected at the center. At that point, we call each of those halves of the X a sister chromatid. So this sister chromatid would have the same information as this other sister chromatid. And that's what we're going to send off into G2 phase and then into mitosis. So we started out with a single sort of stick or worm-like unreplicated chromosome. After S phase, we're going to have these little X's because we've doubled that information. We've made an extra copy of the DNA, but it's still kind of stuck together before we pull it apart during mitosis.